This lesson has to do with drawing a sarcomere, understanding its parts, as we, in the AP1 lecture for Chapter 9, Muscle Physiology, to understand the makeup of the functional unit of skeletal muscle known as the sarcomere. As we start off, the best place to start off in the middle is a thick filament portion that's in the center of the sarcomere called the M line. So we're going to draw it fairly thick. And in the center here, the M line, made mostly of myosin. And the M line is kind of the backbone for this whole structure. Now, when we continue taking our M line, let me write it down so you guys can see that, M line. The M line, we will have coming off of it several lines of myosin that are also thick, that will come off from equal distance on either side of the M line. Now, these are made up of myosin as well, and they are thick filaments, and this is important for us to remember when constructing a sarcomere. Now, on them will be heads of myosin, the myosin active heads that will bind tropomyosin when calcium enters in from the terminal cisternae of the sarcoplasmic reticulum, which you guys have learned about in lecture already. So we need to put this together with existing skeletal muscle physiology, cellular anatomy that we know about for these little heads as they will bind, causing what we, in what process we know as sliding fiber theory. So these need to be all placed on there, these little active heads that will bind to the tropomyosin, to the G-actins found in tropomyosin, and this will cause them to actually flex. And what we can see here now is a small part of what we know is the H band, but we can also see a bulk of the A band, and we've got to put this together. Now, as we go into this, I like to, for simplicity's sake, take my thin filaments, my actin filaments, and put them between each and every single thick filament before I construct my Z-line. Number one, this makes it neater, but number two, we want to try to get to understand this cellular component, this protein component of the muscle cell. We really have to be able to construct it, and this is very helpful. Now, once you have built this up, take time to draw the Z-lines at the ends of your sarcomere, and note that from here on out there will be another sarcomere on the other side, and this creates those striations that we notice in skeletal muscle tissue. So here we have our orange lines. These orange lines, these are comprised of thin filaments of actin, and actin, you know, it has troponin, tropomyosin on it. This is between the myosin heads, we already kind of know, and so there are a few things we need to add in here. As we have drawn out a sarcomere that is pretty well intact so far, I will add a couple extra things to it. But right now we see the M line, then we take our red and we go into here, and you see from here to here the H band. And your H band is this zone of completely nothing but thick filaments. Now there is an A band that sits here. Between the A band contains both thick and thin filaments, and then there is a nice area here, highly visible on this diagram that I've drawn for you, called the zone of overlap. And the zone of overlap contains both thick and thin filaments. So the zone of overlap, and we can see that nicely on this sarcomere. Now, just as a reminder of how to draw that out, what is going on there, the M line, the Z line is on the other side, the Z lines, and the A band, the H band, the M line, because the questions I want you to be able to answer about this is particularly what kind of fibers make up this, what be able to make up that. Be able to draw this from memory on your exams, and you can get your replacement points for that. But this lesson, I'll be able to help you in drawing this so you can see that, as you will also look at other lessons on neuromuscular junctions that are very similar to this. But I do hope you find this helpful, and that is the lesson on how to draw a sarcomere and the sarcomere portions. And so I'll go ahead and stop recording now.